Is my audio functioning?
Yeah.
Okay.
Yeah, it's recording. So, okay, so today, the purpose of today meeting is for us to have a discussion about our next paper for open source mycetoma. So we'll go through firstly just a quick introduction from each one of us. So we have an idea of like who everyone is and what they're working on. And then we will have a synthetic update. So any progress we've made so far, and any plan or now delayed plan of progress for synthetics to we can come back to when the pandemic is over. Also biological results, any discussion on that, and then we'll talk about the scope of our paper. So what how we would define the boundaries of our research for this particular paper. Then we actually talk about this specific of the paper writing, so including how the different section and also how we can collaboratively write this piece of this article together. Then deadline, and then we also have any other business at the end as well. So I guess we'll start with the introduction. So I'll probably just direct that from who I can see here in my view. So okay, I'll start. My name is Hung, and I am a PhD student. So I'm working on organic chemistry and also science communication as well. So I work for my supervisors are Alice Motion, who's also in the meeting today, and Peter Relich as well, who are also in the meeting. So I started my honors with open source mycetoma. So I was working with MapTop, and now I kind of expanded my interest towards science communication as well. So you can either, I guess you can do a quick introduction in the chat, or you can also just unmute yourself and have a chat as well.
Hi, this is Ben from the NDI. Good to see you all.
Hello all, it's Matt Todd, calling in from London. It's great that we're having this meeting, because I think, as you can see, there's lots of people on the call and lots of people interested. So I think it's a great idea to connect. Good morning, everybody.
Good morning, my name is Wendy. I'm heading the Mycetoma Lab and SMC. I hope you can all hear me, because for me it's echo on the line.
Hi, this is Wilson, and I'm also from the Erasmus MC, working under Wendy at the moment. And yeah, I was involved in the first paper with you all.
Hi, I'm Kimberly. I'm new to this project. I'm with Alice, University of Sydney this year, so just here to check it out and info about what you guys are up to.
My name is Anthony, but I usually go by my nickname, Mandrake. I'm just someone who's self-taught in biochem, got interested in it, and I've been helping with designing different compounds and stuff. My main interests are in fungi, both pathogenic and non-pathogenic, and in amoeba, both pathogenic and non-pathogenic.
Hello, this is Dmitry calling from Lithuania. I'm a PhD student from UCL School of Pharmacy.
Hi, everyone, I'm Alice Motion, calling from Sydney. Nice to see so many faces and names, familiar and unfamiliar. Working with Peter and Hung, and I've been working with Matt for a very long time too, so nice to see all of you in this meeting.
Hi, I'm Peter. I'm also in Sydney, and long-time collaborator with Matt and Alice, Hung's auxiliary supervisor. Excited to be part of this project. Nice to see you all.
Hi, I'm Mickey. I'm working for Wendy at Erasmus as well. I just started my PhD and I also was involved in the first paper.
I think that's everyone. So thank you everyone for sharing a bit about yourself. So the next part will be the synthetic updates. So I guess I'll start with what I've been working on during the past month. So here are some of the compounds that I have made or was trying to make. So basically, I'm trying to work on a few different variation of the phenylalanine analog. So changing the pyridine section here into remove the nitrogen to make it a benzyl group, and then switching some of these aromatic rings into aliphatic rings, and also just replacing a ring with just an aliphatic chains instead, and also ring lock as well, to varying success. So the black compounds are what I've already complete. So I'm actually working on the orange one, so almost to completion for this one before the lockdown happened. So I was just about to finish this. I've scheduled for this one. So and then this guy here, I was having some trouble with that. So I actually have just paused, focus on sort of like pause that and just focus on something else, the other compounds instead. So that's what I've been doing. So I guess.
So, Hung, sorry to interrupt. Is that the targets you've got there? Are they on GitHub somewhere, or is I mean, particularly the orange ones? Are they on GitHub somewhere to say that you're doing that?
I think there should be a post under my issue, and it should be on that. So this is the list that I've been working on. I think I have one picture of this. I'll probably put this particular one on. This exact version is not on that yet, but I'll put that on there.
Great. Okay. I mean, it's one of the recurring things, isn't it? That we just it's always quite difficult to have an up-to-date list of who's doing what, and this is one of the recurring things of it in this project, is to make sure that we can have a kind of up-to-date list. There's the list that Ben curates, which has the compounds that are being made as far from the his open synthesis network, DNDI's open synthesis network, which is very useful. And I think we've been trying to add compounds that we've been making over here to that list. If that's the best way of doing it, then great. I mean, we can have a discussion about this later on now, but it's one of those things which is always a difficult thing to do, is have a curated list of which compounds are being made, which ones have been made, and all that.
Yeah.
Yeah, I'm happy to throw those onto that list if you want. Just send the list of compounds to me. It'll take a couple minutes.
Okay. Yes, I'll do that. Thanks. Perhaps Matt, can you continue with the progress over in London?
Yeah. So I think Dimitri may be able to share a slide which has the compounds that he's been making. And the recent structures, which I think he shared, which he sent to Wendy. Dimitri, do you have the slide that you can screen share?
Yeah, sure. Good morning, everyone. Just a second. Can you see the slide? Oh yeah, you can see that.
Not yet, but hopefully.
Can you see the slide?
Not yet. I think you have to click on the little screen and then click the share button, and then it appears. There's one last step you have to do.
Can you see now? Because I'm not sure. I have some confidentiality issues with my computer. I think it wasn't like set up properly, so that's why. Can you see now?
No, sorry.
No?
What's the picture that you've got there, isn't? Ah, there you go.
Yeah. Okay.
Yeah.
Yeah, so I'll give a quick just recap of what I've been doing. So basically, the first scheme refers to my GitHub update that I did recently, just maybe like a month ago before the closure. So I did like 12 analogs, including 2 positive controls, and we sent that to the Netherlands for biological testing, and that has yet to be evaluated. Also, before the lockdown, I was planning to do 2 more analogs from the Austin list, the blue ones on the right-hand side. In addition to that, I also we diverged our series with new AutoXime series. So basically, the biological results that we got from one of the previous screens, the BS0269, showed very good results, and we basically decided to work on a new series based on that result. So as a base, we'll be using a perfinox, which is a known pesticide, so we expect it to have positive results. And then basically we will elaborate on that AutoXime motive to make more analogs, but that refers to the new series. Of course, with possibility of making more analogs, maybe from the Orson list or something like that from the previous series.
That's great. Thanks, Dimitri. The aim here is to, as you can see, to keep the nitrogen and keep the halogenated ring and just play around with the nitrogen-based substituent. Because of course they all come from a common precursor, which is the same precursor as the one that Ben made in quantity for the Open Synthesis Network project, which is good. And in the course of doing this, Dimitri found that if you want to try and put on I think a nitrogen-based substituent there, so if you're using a nitrogen nucleophile, then in some cases the addition of DMAP helped with that reaction, which was useful. I think Dimitri's posted about that. Yeah. And that in some cases that bottom left compound was no good, and we think that's because of a steric problem rather than anything else, because the less encumbered nucleophiles work. Okay. And then the bottom right compound there, this other series, just leapt out of the previous data which we've obtained in the project from Wendy's screen, which showed that those compounds were really quite potent, but also quite synthetically accessible.
The green one is the active.
Yeah, and the long alkyl chain. The long alkyl chains seem to be needed because smaller, less lengthy chains there were inactive. So we were going to just play around with that for a little bit and check that those are active, and then check what can be done there, just to sort of follow all lines of inquiry, basically. But yeah, that's where we're up to.
Then we will have like the third positive control, which is perfinox, because it's one of the precursors of the series, and then we will elaborate on that with further analogs.
So hi, this is Ben. This is really interesting, particularly the fact, as you said, that it seems that the length of the alkyl chain might play into potency, because that ties in maybe with this idea that there's a physical chemical property barrier to activity, and that if it's too short, it's not able to get where it needs to go. I'm just looking at those structures. Those are almost certainly CYP51 inhibitors as well, right? Given that they have an unshielded pyridine.
I believe so. From one paper I found, they said that they were CYP51 inhibitors.
Yeah, it's almost certain. So I was just wondering whether, did these come from the original screen of the malaria box and pathogen box, or has it come from the recent pandemic response box screen, Wendy? Do you know?
It was the original.
It's from the original. Okay, cool. Because I think one thing that we should try to do, if we can, is identify a series that is non-CYP51 inhibition. That would be a real boost to the project, I think.
There were some discussions of possibly doing that. I know there was a kinase, DAF1, that was proposed. I know it's used in commercial pesticides as a inhibitors of a certain kinase, a fungal kinase. There aren't many. I don't think there are any fungus drugs that, well, except for the echinocadins that shut down everything but CYP51. Everything else hits it.
No, that's not true. Olorafim also has a different target. It's
Oh,
now
yeah.
in a clinical trial. It's a DHODH inhibitor.
Oh, yes, I remember you talked to me about that. I apologize.
And terbinafine is also working on the ergosterol pathway, but on a different gene. So that also has a different target.
Correct. I apologize.
Okay,
did miltefosine work? I don't think it did, right?
I have to look into the data. I don't know that by head,
Okay.
actually.
I don't think it worked.
Yeah, so basically that's all I wanted to share with you. If you have any questions.
Yeah. So is there any more question for Dimitri? Okay, so
so soon 2 more will be added, and definitely a few more from the Aldoxine series to come as well. So that's like in the nearest future for my synthetic work as soon as I come back to the lab.
So I think it could be really helpful, again, as the commenter hung, if we can get a list of the compounds that you intend to make and it's constantly updated via GitHub, we can then try and store that in a single location. I'm happy to host that in the way that I've been doing for the Open Synthesis Network, or we can create another repository somewhere, just somewhere we have a list of everything that has been, is being, and is intended to be made. So we're tracking everything in a single location and kind of get into the habit of posting those new compounds somewhere. And as long as there's, I guess, if there's someone who can be accountable for updating that list, which I'm happy to do, or it should be as simple as just posting a smile string in a comment on GitHub as to, here's a new design, here's new molecules I'm working on, and tag whoever the accountable person is, if that's me, and then hopefully that person, maybe me, gets around to actually updating the file.
So is it better to have like a new issue on GitHub, or shall we stick to the network?
Yeah, I guess we could create a new issue on GitHub where there is just, post new designs, post all compounds here. I would try to avoid having too many different threads with different sets of information. So maybe if there is one thread that contains, all of the information on what has been made, what is going to be made, what's being made, and every time there's a successful synthesis, put a comment in there saying this has been made. Every time there's a I'm now making this compound, or I thought about this compound, put it in there so that we can have kind of like a single track. That might be the best, I guess. That's my view. I don't know if anyone else has other thoughts on that, but I think it would be
sometimes it's a bit problematic,
important to kind of track it.
One or a few at a time, and so I thought like maybe initially to update like as a large batch, so it would be like easier for you just to update the list rather than like give you like every week like 2 or 3 more compounds. But maybe like in the new thread we need to update it like in real time, just to make people aware like what's happening.
Yeah, I think this is an issue we always face, and there's no quick solution for it, but we can gradually try and solve it. The updatable list of things that are in process is always a tricky one, and we're great to solve it one day.
So I'm in theory working on this for the Open Synthesis Network at DNDi over the next year or so, trying to come up with a website that works for us that does this, which in theory can then be used for everything else. Luckily, I'm not sure if everyone has seen this, but the Open COVID project at Diamond that's being hosted by Postera, they kind of basically have nailed this. I haven't got around to writing to them to find out what they've used, what the API in the background is, but I think that they've come up with a really elegant solution to this, where you can just upload compounds and it gets put into a repository.
I've contributed a bit to that. They said everything's custom written, I believe.
Mm-hmm. Yeah, it's custom written, so I just need to write to them and ask whether I can get a copy. So we'll see, but I think that works quite well.
Okay, yeah, thank you. That sounds really, really great. And also, I guess in the meantime, we can perhaps create a new issue in GitHub, and then we just leave that for just progress instead of, and then we'll have discussion and other things in other channels, but just have one that's dedicated. Is the list that we're doing now is what we're planning to make, and that could also be a potential solution as well.
Yeah, I think that'd be really good, Ben. I think your suggestion's awesome and excellent. I wonder if, in the meantime, the color scheme, for example, that Hung used in his previous slide, if people had one image that's of the drugs that they've identified that they're working on, the molecules they're working on, and then if they change the color of them and just re-upload the image, it even gives us just a quick sense of what's happening and where the problems might be. So we could maybe do a little bit of both.
I think Ben's list has already has color coding, so I guess we can all work to hit standard instead. I think that would be a better.
I like Ben's color code too. I really like his list. I just mean if people wanted to upload a snapshot while we're waiting for Ben to do this, because it puts a bit of a burden on Ben too. So I think Ben's idea is awesome, but just wondering if we could try and make sure that we know where we are as a snapshot too.
Yeah, I'm just going to second that. I think that would be super helpful. Like, I really do like the way that, guys, there's this format, like as we see on the screen here, where you've got everything and different colors. I guess the problem with the way that I'm posting them is that it's done really quickly, but it's done through Stardrop, and that's obviously a costly piece of software that luckily I have access to. But if people can do, as Alice just suggested, an update with what they're currently working on as a single screenshot colored the way that you do it, that'll really help me to make sure I don't miss anything, because then I can just go back through and I can spend half hour making sure.
So does that imply that every single time when we do something new, we need to upload it like straight away to GitHub, like as a new image or something like that?
I think it would be great, Dimitri. So if you think about this image here, so obviously your top ones, you've sent those all for biological evaluation. That's great. But it would be something that would be as quick as if you had a list of all the things you were working on, just when you finish one, you just have to change the color and save it as an image and upload. So it shouldn't be too painful. I was just trying to think of something that we could do really quickly that would just have a snapshot of that information without even having to send an email or anything.
Yeah, I think that will work.
And I think so I think that's. Is there any other synthetic updates that we need to know about?
Those are the main ones, because I'm doing an upgrade report currently. So I finished like those series and then now currently doing the write-up. But at the same time, as we come back, I will finish all of the DM27, DM28, and the Alexan series as well. Actually, Alexan series has been started like some time ago, so we have some results, the intermediate, but it needs to be finished. That's basically it.
Just on the Seven Oaks school as well. So the Seven Oaks school are making one compound, but because of the shutdown, they've had to pause it. So they haven't quite got to the finishing point yet. But hopefully when they reopen, they can resume the project, but we'll have to wait and see.
Okay, that's great.
Yeah, same situation for the few participants in the OSN who were working on this. I think that there was some enthusiasm from the team at University of Washington to make some compounds using good chemistry, but I'm guessing, I haven't had an update from them recently, that that's either delayed or cancelled because obviously classes are out and everyone's working from home.
Yeah, I saw that they did like quite chosen a large list of molecules to make, like I can see like from the OSN list, but we haven't heard like from them for a while.
Yeah, so that was that. It was a teaching lab that was scheduled to happen this semester, I think, or if not, a little bit over this semester and a little bit over the summer, with quite a few students. But I'm guessing that's off, unfortunately.
Okay, so I guess we'll be moving on to the biological evaluation. So if Wendy or everyone from Wendy's team can give us an update to.
Yeah, I will be giving a short overview. Just let me figure this out to put it on screen. Is my screen shared? Yeah. All right.
Yes.
So for this, so after the first paper, after MycetoS 1, we've received about 73 isolates from Epichem. That's the phenarimoles batch 2. And then Hung gave us 16 compounds or analogs. And then I've also received 7 from the P series. We've received that last year. And that we have done initial screening. And then we moved on to look at IC50. So from the Epichem batch that Ben gave us, 11 worked. From Hung 16, 11 worked. And from the P series, which not worked, but we tested. So 11 from Epichem, 11 from Hung, and 7 from the P series we've done at MIC. That meaning that we tested on our main Madurella isolate, MM55. Sorry, this is a bit confusing. And then from testing one fungal isolate, we went on to test a range of 10 other Madurella mycetomatis isolates. So that's the MIC. And from these, we see that 7 from Epichem, or the phenarimoles batch 2, inhibited the fungal at a low concentration, and 6 from Hung's, and only 1 from the P series inhibited the fungal isolates below 10 micromolar. And
sorry, Wilson, can I just ask what's the P series?
The P series are 7 analogs that we received last year. Their name starts with P. P4 something something.
Did they come from us?
Yeah.
Oh, okay. Does anyone? Which compounds are those? I'm not sure I've seen that phrase before.
Open Synthesis Network had them entered in as P4 something.
Yeah.
Oh, is that what it is? Okay.
Yeah, if Ben can elaborate.
They're on the list, Matt. They're on the list. You under the codes.
Okay. All right. So
yeah. So we tested these, and our in vitro testing came up with 10 that worked under 10 micromolar in 10 different Madurella mycetomatis isolates. And from now on, we'll be choosing 8 to do in vivo work. 8 because 2 of them worked above 4 micromolar, So we want to reduce it as much as possible, and only test analogs or compounds that can inhibit the fungus below 4 micromolar. That makes it easier. I've also this is the data, the growth percentage for all that we've tested under 100 micromolar. You can see the gray is the first series, phenomenal series that we received up to the first paper. Black is from the Epichem. The red is analogs from home, and blue, the P series. You can see that under 100 micromolars, only a selected few was able to inhibit growth to under 20%, or inhibit more than 80% growth. And you can see that lots of Home's compound actually works really well, except for 6 of them. So I don't know what you're doing, Home, but it's quite good. Then we looked at IC50. This is only for one fungal strain that we tested, and again we see that only 3 from the black one is promising, and none of the blue ones, but about 6 from Homes is quite promising, and that 6 will be move forward in our in vivo work. For what we still have to do, there are some analogs from Ben's Epichem series that we still have not tested them in vivo, in vitro. We've missed them somehow, and this will be done once the lab is, once we're allowed to go back to the lab. So there's about 5 of them. We can finish them in a month's time.
Just to mention, we also received just before the lockdown some compounds, of course, from Matt's lab, and the compounds of Hung are also probably going to be sent in a short while, and those will be included as well, of course, for the MICs.
Yeah.
Yes, good. I'll get to sending my next batch over soon as well.
So yeah, this is not including what we'll still have to receive. But for what we have now, the next, also we have about 8, well, let's say 10 on average to do tests in vivo from 6 from Homes and 2 from Epichem series, and maybe 1 or 2 depending on the missing in vitro data. And we are hoping to start up the in vivo work in the next month. Hopefully we are allowed to do that. And we may expect completion of these somewhere late autumn. That's when we'll get the complete data, and that's when we can upload everything.
Just
so for now, these.
That autumn. That's in the. That's which month is that? Well.
So our in vivo larvae work, we can only do it in summer. So depending on when we get to start up, we have to rush things. We may, it really depends on when we can finish and when the larvae season ends. Let's say September latest, or October. It might go into October. That's the experiments. But then the grain counting, the histology, that might take a few more weeks to a month. So I'd say about, on the safe side, October, November.
Okay, thank you.
Yeah. Yeah, that's it for my side. Yeah. Any questions?
I just would like to add that our larvae season, unfortunately, is dependent on the season. And also the larvae are raised in Italy. We already got word that we are able to get the larvae. And we also got word that we are allowed to start this up, but due to the slower culture times of Madurella mycetomatis, and that we need to get the larvae, by the best thing we could do, we can start within 2 weeks to 4 weeks for the first experiment. But the first experiment always is re-evaluating the model to see if it does what it's supposed to do. And of course, with the analogs we test in vivo, we are also including itraconazole because it's the current drug of treatment. And we were also considering using 2 of the previous EPL compounds, one which has a very high survival and one which has a very low survival. So these were the controls we were thinking of. That's just to demonstrate that the model does exactly what it did in the previous paper and for comparison.
Yeah, and I think in my post for the next batch, I also include 2 compounds that I previously made as well, also as a just to double check that it's still working fine.
So just a question, Hung, how many compounds will you be sending over in the next batch? I can't hear you.
So 4 new ones
yeah.
for retest.
Yeah. So 4 new ones.
Yes.
Okay. Yeah. Thanks.
Just for the tracking which compounds are which, I just put a link to the spreadsheet there of all the molecules in the project. And just want to encourage everyone who's making molecules or has made molecules or has tested molecules to try to be sure that the compounds are in that sheet, because it's sort of the master list of everything. There's a bunch of stuff there, but the third tab on there are the MycetoS compounds. And so, if something's made or tested, if it can go there with the ID, that'd be really good. And there's a notes column which can be used to install a link to, where the molecule is made or suggested or evaluated and so on, so we can track where things are. We had talked at the beginning about having MycetoS IDs for compounds. We haven't done that in the sheet yet, but we can at any point. But just as long as the codes are there and we know what the molecules are, that's good. If people can just check that molecules that they've made or evaluated are in that sheet, that'd be really helpful.
Thanks, Matt, for that. So, is there any more question for Wilson and Wendy? If not, we will move on to our next item, which is the discussion of our next paper. So, well, firstly, how we're going to write this together, and also the different section of that as well. So I've already uploaded a Google Docs of the experimental method that I've put together. Perhaps we can work from there, or does anyone have a suggestion for that?
Maybe it's good to first discuss what we should put in the paper. Should we start with the EpiChem library? Should that be included, or should we only include the synthesis, or what's the idea behind it? Because that also depends on what kind of figures we're going to make, etc.
Okay, yeah, that sounds good.
Because for us, we're not chemists. For us, the Fennery moles is the whole. So that's EpiChem, including the things synthesized via the MycetoS. But I'm not sure if that's for the chemists the same thing as for our simple biologists.
I think, Wendy, I think it is. I think all of the compounds that have been made recently so far have all been phenaramol-like. I mean, that includes the compounds that Wilson flagged up as P to P-like. Those are phenaramols as well. So they're similar to the EpiChem. So I would say that it's possible to have a single paper that focuses on a single structural type. So I think almost all of those compounds that have been tested would be covered under that umbrella.
Yeah, so is the aim of the paper to talk about the exploration of the phenaromoles and recent data and so on? If it is, then we will definitely need to be waiting for some in vivo data, right? So that's what Wilson was just talking about.
Yes, yeah, unfortunately, the in vivo data will not go faster as it goes because there are many experiments. We have to repeat several of the experiments. We always do it in triplicate, and one experiment already takes 4 weeks from start till end. But we could, what Wilson said, we usually also include grain counts. But last time they were not that informative, so we could also decide only to go with survival curves and to only do a single histology point or something like that, but not counting. That will reduce the amount of work, but it also will lose a little bit of information we had in the first paper.
It also depends a little bit on the theme of it, right? Because I mean, one of the most interesting things about the first paper was this correlation that Ben saw between log D and in vivo potency, right? Which we can talk about in a minute. But that was a big issue. And so if you want to try and add an interesting element to the paper, we should be tracking that. In other words, compounds that are made and evaluated, we need to be aware of what their log Ds are, so that we can see if new compounds back up that idea. In other words, even if we don't discover an amazing new compound for this paper, we can still focus in on that relationship.
Yes, so I've my compound has variation in log D, so that could be one thing that to look out for as well. Okay, so so paper planning. Well, the first item we have is journal selection. So I'm not very experienced in this, so would love to have everyone who's done this before to actually give a suggestion. I think since we've done this in PLOS NCD last time, we could look into doing that again if possible.
But I think it's going to depend on what we put in there, I think, and what the message is, if it's chemical or more biological. So I think that we're going to have to. I mean, it has to be open access journal, right? But I think the journal choice has to come later once we're more sure about what's going in there.
I agree, because I don't think PLOS will accept a very synthesis-like paper. But we can always opt for the open access with any journal.
So just quickly while we're talking about that, so yesterday Wendy and I submitted a proposal to look at the grain penetration side of things. So which molecules get in to the grain and which don't, because it's the central question that we've got. And so we're going to try and generate some resources for that side of the project. Part of that is trying to figure out by direct measurement of which molecules get in and which don't. Wendy, do you have a sense that any of that stuff could be attempted in the shorter term to see if we can take molecules that work well and badly in the in vivo assay and try and measure if they are grain penetrant? Is any of it doable in the shorter term, or does it kind of depend on us getting the resources in the grant?
Well, it's a little bit difficult because we're currently still shut down and we didn't have an official date when we are opening yet. Because of the seasonality of the larvae, we are allowed to get into the lab for the larvae, but we're not allowed to get in the lab for all kinds of other things yet. And I don't know when that will happen. The lockdown in the Netherlands is still till the 20th of May at the moment. And we could develop an in vitro model which produces grains. We also have a few isolates which actually do that naturally, a little bit grain-like structures. but how predictive it is in the for the larvae model, we don't know yet. So I would love to say, yes, I can do that in a very short notice, but two things are playing a role here. First is lockdown, and the second one is I only know when the model is right when I can compare. So I need to do the larvae model anyways, and the next step would be to test the molecules we tested in the larvae in this new to be developed model, to see if it's predictive, yes or no. Also, NMR to do that on histology sections or something like that. It's not up and running at the moment. I need to get some context to get that done. That's something which might be doable when we have the larvae. But again, yeah, the larvae are difficult to penetrate. So it's not as easy as it sounds.
No, it doesn't sound easy. But I just think I'm thinking about the paper. If we're thinking about a paper, then either it has to be something where we blitz the SAR and we generate, I think, a lot more data points than we have already, just measuring in vitro potency, or we try and do something which has a biological message and a lesson about what kind of molecules we should be making. I think either of those has an easy sell. But at the moment, you know, we're not quite. We don't have a sort of a set of data which I think a lot of people in the field will be interested in yet, because we don't have something phenoremol which is particularly, you know, highly active and novel yet, compared to what we have, and we don't have something which is a clear picture of what we need for the molecular design.
Now, I think if we have the larvae data, we only looked at log D. I know that at GitHub, some students also looked at other things, other properties of the molecules. We didn't publish on that yet. Maybe when we have this larger subset also in the larvae, we could include those type of analyses as well. So that makes it also a little bit more broad than it was in the previous one. And we simply have a very large, much larger collection now. We could also, since we still need to do some in vitro susceptibility, do a second batch of epichem so that we have a little bit more mass of molecules, if that's helpful. But we could also wait a little bit on the larvae data. But the risk there is that if we want to test any additional molecules for the larvae data, we really need to know now which molecules we are testing. Maybe one or two can be added during the season, but if there's too many compounds to be added, I'm fearing that we will not make it within this season, because the season has already got short due to the COVID outbreak.
Just on the other molecular descriptors thing, I had a student, Rosalind Chan, who did a lit project on that aspect of things and looked at the some of the other molecular parameters. And I just need to check whether I can share that with everybody, because it was under assessment at UCL. But I think it's been done now, so I'll be able to share that, where she tracks some of these things like molecular weight and approximations of molecular size and whether that mattered in terms of grain penetration. So I'll try and get that posted as soon as I can.
There's an issue on that on GitHub as well.
I think that also ties in with the next item that we have for today, which is the scope or the boundary of what we need for this paper. So it's a little bit more complicated now because everything is shut down or delayed. So so initially I was thinking of either a deadline or a number of molecules as a boundary. But if anyone has a suggestion, that would be greatly appreciated.
Well, for the molecules we screened so far, we now made a list which we are going to test in vivo. I think it's good to do these in vivo and set the deadline of the paper in November, for instance, because then we would have all those in vivo data. We will add gradually on the in vivo data. We can write all the way up to the in vitro screenings, of course, because we already have that data. And when the data comes in, we use GitHub to share the data with you. And we will make the figures for the and that part of the paper. Of course, we only can calculate the correlation, or at least Ben probably can, after the in vivo data is there. So last time we correlated log D to percentage survival. That's one of the first parameters we will have. Of course, we have to check if one of the Fennery moles is toxic for our larvae, but we can do that simultaneously. So any compounds which show some toxicity will be left out of the equation, because it's not a fair equation if the compound itself is toxic. But at least we can do it on all similarly in the same time and real time, because usually we test them batch-wise. So maybe the first 6 compounds will get in before the other 6 compounds or something like that. But that's, yeah, that will give us a sort of boundary and also a deadline.
Okay, so I think that's a great idea. So we can we can set the paper deadline to November, and then I guess however many compounds we will be able to be synthesized and tested by then will go into the paper. Does anyone else have any other suggestion for this?
I'm just curious, are we going to still continue to expand on the SAR tables after the paper is published? I assume so, yeah? Like kind of fill in the blanks in the SAR?
Yeah, I mean, it's an interesting one, because of course there's no hard stop, right? I mean, the data accumulate and the picture changes, but there's always new molecules to be made, and I guess aiming for a paper might help us to make a decision about whether we're going to actively pursue phenaromoles more, which would be a useful point to get to. Just because obviously this isn't the only series we could be looking at. And you want to finish a piece of work, but at the same time you want to keep an eye on all those new things that came out of the pandemic response box, and a DHODH inhibitor idea. So I don't know. I think yeah, we have to investigate more and publish, and then maybe towards the end of the year we can be making the decision about whether we're going to be actively plugging those SAR holes. Which means, of course, the more compounds we can make in the series, the better before that deadline. And I don't know if anybody had any unannounced or unexplored avenues for people who could make compounds for the series, to sort of contribute, because we obviously, Hung's making compounds and Dimitri's making compounds, and people on the Open Synthesis Network are making compounds, and that's great. Are there other people who might be able to be encouraged to make a molecule or two? Does anyone have any leads there? In the time that we've got until the next paper, anyone got any leads for people who might be able to chip in?
So we were planning on doing some stuff with some school kids coming into the uni. The problem that's the same problem for everyone. Everything's looking so uncertain that I think that's probably out of the window now, because lab classes have not been running this semester at Sydney, which means that any time, the only way we can get the undergraduates through their lab classes is going to be to, I think, run multiple sessions so that they can work in a socially distanced way, if that makes any sense. So unfortunately, that was our plan. We planned to get some Breaking Good students into the uni this year as part of Kickstart, but now we're running Kickstart online. So our aims have changed from synthetic projects to more computer-based projects. So just wanted to update. I know it's not good news, but just to let you know we were planning something, but looks unlikely now.
And so what about the idea that someone just mentioned about maybe trying to secure a few more compounds from the Epichem library that we haven't looked at yet? Is there Ben, do you think there's value and possibility in that?
Yeah, maybe. what I would suggest would be to, I mean, we can do that now, yes. If the testing capacity is there, then maybe then we should do that. But maybe we want to wait until we've got some sort of stronger hypothesis on what kind of molecules we want to make, what kind of physical chemical properties we want, and then take the compounds at that point. There's no reason why we can't do both. So yeah, I mean, we've got the full list, right? The full list of the Epichem compounds, I think 700 or 800 molecules is available, I believe. So if there are compounds that we want to ship out to have tested from that list, we could do that. And that's kind of a theoretical exercise to identify those, right?
Yeah, it's a good lockdown project, right? Because you could filter all those compounds on log D and get a range.
Yeah, I mean, the issue I see there is that I see a big difference between plugging SAR holes and driving the project forward. And what we should be focusing on is driving the project forward rather than plugging holes, because the holes can be inferred from the data that you get. So, I mean, we can do it. I'm just not convinced it's going to bring a huge amount to driving the project forward compared to the data that we've already got. But if the capacity is there to test and it's generating more data, then yeah, let's do it.
And maybe it's an idea because we're thinking of not only Fennery moles, but what we learned from the Fennery moles of the log D. I'm not a chemist, but I assume that also is true for other chemical compounds. So maybe there are similar lines we could take, and if log D for the Fennery moles is important, wouldn't it be a suggestion to see for another series where we can repeat some of it and see if it's true for not only the Fennery moles, but also for other compound series?
Absolutely. I mean, when you go from one series to another series, those rules are not, they don't transfer clear cut, but usually the general trends could translate across if we are indeed dealing with a activity that's driven by access to permeability or access to a particular part of the structure or a particular part of the pathogen. But I think that that's kind of similar to, I mean, I think that the proposal that we've made in this Wellcome Trust grant, where we're going to do that exact study, but rather than looking at fungicidal activity, we're going to just look at penetration into the grain using dyes. I think that that is a more robust interpretation of the nature between, for example, log D and permeability than the one that's inferred from looking at the relationship between log D and efficacy. So we could do that, and we should certainly do that on other series if other series have been identified. But we'll be able to do that with a lot more confidence after we've done it with the dyes as per the Wellcome Trust proposal. So I guess the question would be, do we do it now, or do we wait until we see if we get the Wellcome Trust money and whether we do the Wellcome Trust dye proposal first, and then use that to guide the design of compounds in other series? Does that make sense?
Yeah, I mean, I think that the log D filter could apply to the pandemic response box hits in terms of triaging which ones we might be most interested in looking at more and or in vivo, right? Because there's a huge range of molecules there. And it would be interesting to pursue a few of those. I don't know how, but to use that filter there would be pretty useful.
Maybe,
just in terms of, sorry, carry on, Ben.
I was just going to say maybe, but I feel a little bit, I'm always hesitant to apply physical chemical property learnings across different chemotypes. I think that they can tend to be quite robust within a chemotype. Across a chemotype, I'm less confident about doing that. We might risk missing something. I would prefer to, for example, take the most promising hits from that pandemic response box screen, regardless of log D, explore around each one with a set of 5 or 6 or 10 molecules with different physical chemical properties across each chemotype, and then use that data to determine which series we focus on.
Yeah, so maybe Wilson can add a little bit on what we have done so far with the results we had from the pandemic box.
So the pandemic box came with 400 compounds. We tested them initially at 100 micromolars and 25 micromolars. And from there, we went on to test IC50, one Madurella mycetomatis isolate, and then did an MIC with 10 different isolates. We finished the MICs, and we have came up with about 12 to 14, I don't know the precise number in my head, compounds that were able to inhibit growth under 10 micromolar. And part of them, we see compounds from the benzimidazole series. And a few of them are included as well, like olorofim and ravuconazole. So what we have to still have to do next is also the in vivo work for about, like I said, 10 to 12 of these compounds. But like I've mentioned, I think one of the interesting compounds that we can look at is the benzimidazoles. And I think I've also posted a message on GitHub regarding that to see if anyone has a library series of that. And I have no reply since yet. So.
I think those work by inhibiting tubulin.
I'm not too sure about them, actually. I don't have the info in my head at the moment on what they
there must be stores of analogs of benzimidazoles kicking around. I have no idea whether there must be lots, particularly in old dusty industrial labs, that we could try and find. I mean, that's one of the things that made me hesitate about this motif, is that it's so well explored in various areas. But the advantage of that is that I think there'll be compounds that we might be able to get our hands on. I haven't had a chance to look, but if anybody wants to try and have a look at, recent papers on the synthesis of benzimidazole analogs, that would be amazing.
I mean, I would. I guess just because it has a benzimidazole core doesn't mean that every single molecule is related, right? And there are thousands, more than that, tens of thousands of benzimidazole analogs all over the world, but they don't all work through the same mechanism of action. It's all going to depend on how they're decorated and what else is appended to them. So I would say that it's not as simple as just going out there and looking for other benzimidazoles. It needs to be benzimidazoles that have the same kind of functionalization around the core to the one that's found in the hit. So that's going to narrow it down a little bit. And if we would just go and pull back a whole set of different diverse benzimidazoles, we're going to end up screening things which all have different mechanisms of action and work differently and have different relationships. So I think it needs to be quite focused exploration around the hit, which is a much more challenging thing to do through analoguing.
I think the three actives or so they do share a similar substituent, I think. So yeah.
I have to be honest, I haven't looked at them for a while, so I can't remember them, what they look like, but they're really easy to make usually. I mean, if there was a small amount of budget, the best way to do analoguing right now, particularly around simple chemistries like that, is to see what's available from enamine on the real space. So we could do a real space search on those hits and see what the closest analogs we get back from there, and then those are around about $80 to $100 a compound to get hold of, and that's a library of 14 or 15 billion compounds now. So it's going to be far, far more productive than searching the existing databases in pharma companies and whatnot.
I think it's a great idea.
So from the pandemic box, the original benzimidazole that we got a hit from was called fenbendazole. And I went online and looked at other benzimidazoles, and I found mebendazole. From the results we've received, fenbendazole works really well, but not mebendazole. So there might be something there that we could look at.
Yes, thanks Wilson for that update. So I do realize that we are running slightly over time, but there is one more item. So so Mandrake suggests that there is we should have a quick discussion about free living amoeba. So if Mandrake, can you just
this was a follow-up to the idea I had with Dr. Dennis Kyle, if you remember, Dr. Todd, about getting possibly funding from a different source, because there are some small charities available that fund research on these pathogens called pathogenic free-living amoebae. They're mostly CNS pathogens. They're a little bit fungus-like, but they're not taxonomically related. But I figure it might be a way of getting extra funding. They've always stuck in my mind because of, I guess, just because of the personal interest of the story that I heard growing up, I guess.
Yeah, so what's needed?
Well, there is some drugs available now for it, but I guess the idea would be getting something that has better inhibitory concentrations. Dr. Kyle said that he found posaconazole was the best of the FDA-approved compounds, but that's not the standard of care now, so they're waiting to try that, I guess. But it's possible that we can get some tested. It would provide another data point and possibly some funding, because they want to give grants to researchers on these things. They're so understudied.
Yeah, no, I mean, in terms of what we would be able to do, so is the idea, I mean, is the blockage here that we need to ship some actives or a few molecules to him? Is that where we're up to?
I believe so. He has an assay he has set up, but he's probably closed down by now. But he might open up sometime soon. I don't know. It's always been something of interest to me ever since I heard about them. Fascinating organisms. But there's nothing made just for them. All the drugs are leishmania killers or fungus killers. So there's overlap. They have CYP51. They have some fungal kinases. Might be something to side screen.
But I mean, the next step would be that if we think that it's a good idea that the compounds from Sydney and from London could be shipped to him. I mean, that would be the next step if we're convinced that
Correct.
it's
Correct.
Okay, so I guess we just need to have a look at that and decide on what to do. I'm happy to reach out to Dennis about doing that. I think he's amenable.
Yeah,
but it's a good idea to ship molecules.
Yeah, it's 3 amoeba screen, which the Dalmuthia nigliria and Acanthamoeba, all of which are usually CNS pathogens. Pretty low survival rate, but it's gotten better with this drug called miltefosine, which is an amoebicide, leishmanicide, and fungicide. But other than that, it's been pretty grim otherwise, but I figure it is another way of getting funding, right? And it is a way of moving the whole project forward. And they have some slight overlap biochemically with fungi, although they're not related at all.
Yes, that sounds very good. So we can look into that a bit further as we go along. That is all for all of the items for this meeting. So I guess now we should look into perhaps planning for our next meeting, which I guess perhaps should be in whenever perhaps the shutdown is lifted, so we can have a next meeting by then, just to see like what we can do from there. Does anyone have any other suggestion apart from that?
Meeting in 2 months?
Yes, that's what I kind of thought as well. So I think that would be kind of roughly a good idea.
I just had a quick question to Dr. Sand, to Randy, regarding the molecules and the testing. I just want to get clarification. So are we limited to the number of molecules that we can test during the season? Because you mentioned this and I didn't get this. Maybe I didn't get this right. So like, are we limited with the number of molecules that we can test during the season?
Well, only for the in vivo part. So the in vitro part, we can do more than that. Only when we receive molecules relatively late in the larvae season, and they are promising, then I'm not sure that we can add them to the in vivo screen, so to the larvae screen. So for in vitro, that's not an issue. We can screen that the whole year, but it's only for screening inside the larvae. We already have your molecules in the lab, so those will be tested together with the other venery moles. So when we are allowed to get back, and hopefully in the waiting steps of the larvae model, because we have a 4-hour waiting step, we can do the in vitro susceptibility because we're already there in the lab. So at that time point, we are there. But yeah, I assume that the compounds Hung is sending and your compounds, which are already in our lab, those are still be included in the larvae screen if any of them appear to be hits, because we will know that in advance.
Sure, thank you.
I think also it might be good, certainly if Dimitri and Hung, so I think Hung's written up all of the experimental data now for his compounds that he's made. And I guess, Dimitri, from what you were saying, you've been doing that a little bit while you're away from the lab. If we could share the experimental data somewhere so that we have it all together, so that people can add that in as we go along, and maybe starting to write up some of the results and discussion part about the synthesis might be a good way to start, so there's some sections ready for the paper, even if we haven't decided exactly what the story is for the paper just yet. It might just mean that some of those chemistry bits are tied up and ready to go.
So I'm currently writing the upgrade report for my PhD. So basically the experimental part is almost done.
Oh, that's
So
great.
I assume I would be able to use that like straight away, like from my work to the paper. So
That's great.
It will be finished. It's almost done. It will be finished soon.
Great news. Good luck with that report. I know that I'm sure you
And
do.
of course, we'll keep adding the new molecules and the characterization like in full.
Okay. Yeah, that's fabulous. Because I think, there's always there are always lots of challenges about these exciting open projects, and making sure we have all the chemistry data written up. That always helps with pushing forward the publication and things like that.
But of course, we update everything like on ELN as well.
Mm-hmm. Yeah, that's great. But just having it all in one place could be useful, I think.
Like a new issue, GitHub issue.
Or just even in a shared Google Doc or a shared Dropbox folder, put it all together.
Yeah, good idea.
Put it into the Google Docs folder that I've already had on GitHub, so we can just put that together. So I've already got my version up there as well. I still need to be tweaked a little bit, but you can certainly add yours to that as well. Yeah, so I think that that is all of the issues that I have today. So does anyone have any other things that I would like to have to talk about before we conclude today's meeting?
Yeah, on the Zoom chat, it was proposed to be the 1st of July for the next meeting, but I was wondering if it's also possible to have it 30th of June, because on Wednesday I usually have to take the children to school, and in the Netherlands the schools are starting up again. So at this particular point of the meeting, I would be at school.
Yeah, both days are clear. So sounds good.
Okay, so yes, so 30th of June, is everyone good with that? If so, we will schedule that for 30th of June.
The same time work for everybody as well as what we've done this meeting.
Yes, great. So so I'll schedule another one for 30th of June. That's 6 a.m. GMT. So thank you everyone for joining today and discussing about our paper, and I'll hope to see everyone soon.
Great, Hung. Are you able to post the recording and also begin an issue with the action items for people? Is that all right?
Yes. So I'll do that after the meeting's done.
great. That'd be fantastic. Thanks so much.
Thank you very much. I'll see everyone later.
Bye.
Bye.
Bye, everyone. Thanks.
Bye.
Bye. Nice to meet you all.
Bye-bye.
Nice to meet you too.
Hi, Hong. Well done.
Thanks. That was very stressful. I didn't mess up too bad, did I?
No, it was really good. It's really good. These are the things these are the skills